A very good afternoon to one and all. This is Dr. Akanksha, the business student leading to the university. Today we'll be talking about biosensor. So biosensor is nothing but a short form for biological sensor. Basically, it's an analytical device which is used for detection of any analyte that combines a biological component with a physiochemical detector. Now, father of biosensor is Professor Leland C. Clark. It is a device which incorporates a biological sensing element either directly connected to the transducer or some way integrated with the transducer. Now, the recognition in biosensor is basically based on the affinity between the complementary structures, say enzyme and substrate, antibody and antigen, receptor hormone and targets. So basically, the main principle behind this is selectivity and specificity. Here, selectivity and specificity depends on biological recognition of the system connected to a suitable transducer. So we'll be talking of the biosensor, what exactly biosensor does. Basically, biosensor converts your response, which is in form of chemical reactions, into electrical signal. It detects and records and also transmits information regarding a physiological changes or a process which is occurring in your body. It determines the presence and concentration of specific substances in a test solution. And main features of biosensor which we look for is stability, cost affordability, sensitivity, and reproducibility. Moving further to basic principle of biosensor. So biosensor basically involves three major elements. The first is biological recognition element, which is highly specific towards the biological material, analyte, which is produced or coated. Now, secondary transducer detects and transduces the signal from biological target receptor molecule to electrical signal, which is due to the reaction. Last but not the least, after transduction signal is received from biological to electrical signal, if amplification is needed, an amplifier is connected, and then detector will record its reading and display. So moving on to the components, as you can see here, three major components can be seen: bioreceptor, transducer, and measurable signals. Now here if you see analyte, this analyte will be reacting with the biosensor which you are keeping or coating, which usually includes enzyme, antibody, microorganisms, and CDA. Then a transducer is connected, which is in form of electrodes, semiconductor pH electrode, photo counter, or piezoelectric devices, which will measure the signal of this chemical reactions and convert it into electrical signals, which further will be detected. So moving on to the components one by one. So the first component is the biological element, which is used to bind to the target molecule. And it must be highly specific and stable under storage conditions and immobilized. A variety of substance that may be used as a bioelement, but basically most commonly used are microorganisms, tissues, cell organelles, nucleic acids, enzymes, receptor, antibody, or plant proteins. Second component and the foremost important component is physicochemical transducer, which acts basically as an interface measuring the physical change that has occurred in the reaction at the bioreceptor surface and then transforming the energy into measurable electrical output. The third component is detector, which will be taking signals from detector and passed on to microprocessor where they can be amplified. The data can be converted to concentration units and transferred to the data storage device. So the principle of detection basically depends on piezoelectric device which you are keeping as a detector, electromechanical, optical, and calorimetric. Piezoelectric basically measures the change in mass, electromechanical and electric distribution, optical measures changes in light, and calorimetric changes measures in heat. So the working principle, as I discussed, includes analyte basically diffuses from the solution to the surface of the biosensor, where it reacts with the surface biological component of the biosensor. This reaction will change the physicochemical properties of the transducer surface. Now, this will lead to a change in optical or electronic properties at the transducer surface, which will be read by a transducer and converted into electrical signals. So usually enzyme or preferred biological material is deactivated by some usual method, that is deactivated biological material in near contact to transducer. So analyte connects to the object, to the shape, clear and light, and turns into electronic reactions. In some examples, analyze is changed to device which may be connected to the discharge of gas, electron ions. So in this transducer, can also alter the device linked with the electrical changes, which can be changed and measured accordingly. Now here is a schematic diagram of biosensor where you can see a bioelement which will be reacting on this surface, then transducer which is reading these reactions and converting into electrical signal. In some cases it needs to be amplified. So amplifier will amplify it and processor will read that reading and you get a unit on a display machine. And the best example is your glucometer. So how does this work? It involves a quantitative analysis of substance by converting their biological actions into measurable signals. Generally, the performance of biosensor is mostly dependent on specificity of the biosensor and sensitivity of the biochemical reaction. Besides, stability of enzyme is equally important. So an ideal biosensor should have these following properties, which we are discussing here. First thing, the output signal should be relevant to the measurement of the environment, and the functional surface must be compatible with transducer. High specificity and sensitivity is required. Substance sensitivity and resolution should be there. Surface accuracy, speed of response should be prompt, sufficient dynamic range, and last, sensitivity to environmental interference is equally important. So basic characteristics of biosensors which we look includes linearity, sensitivity, selectivity, and response time. Linearity should be high for detection of high substrate, low for detection of low substrate. Sensitivity, value of electrode response per substrate can be detected by your biosensor, so it should be that sensitive. Selectivity, chemical interference must be minimized for obtaining the correct result. And as I told, response time is highly important. So time necessary for having 95% of response should be there in a particular biosensor. Now, applications of biosensor basically includes food analysis, study of biomolecules, drug development, crime detection, medical diagnosis, quality control, industrial process control, biological welfare, aging detection, or manufacturing pharmaceutical drugs. In detail, applications of food industry basically deals with monitoring of fermentation products where it helps in improving the fermentation conditions. Nowadays, biosensors are employed to measure the odor and freshness of food also. Freshness of stored fish can be detected by ATPase enzyme using a biosensor. Talking of applications in pollution control, so here biosensors are helpful to monitor environmental pollution, concentration of pesticides can be measured, and also it has wide application for measurement and control of water, soil, air contamination. Especially determination of pesticides can also be done by potentiometric biosensor. Last, applications in military defense biosensors are also developed to detect the toxic gases and other chemical reagents which are used during the war. DNA biosensor is also used to detect chemicals. So these are a little glimpse of biosensor, its component, principle, and its application. Thank you. Have a nice day.